life embarks on its intricate journey when the reproductive systems of both males and females work together, all under the guidance of the brain. At the start of this biological chain lies the hypothalamus, a crucial region in the brain that begins reproduction. In response to both internal and external stimuli, the hypothalamus releases chemical messengers that travel to the pituitary gland, prompting it to activate this biological process. The pituitary gland, often referred to as the master gland, springs into action and releases two vital hormones that begin the reproductive cycle. FSH, follicle-stimulating hormone. This hormone plays a significant role in germ cell development in both sexes. LH, luteinizing hormone. LH helps initiate the production of sex hormones and regulates the reproductive cycle. In males, the effects of FSH and LH are significant. FSH initiates spermatogenesis, the process by which new sperm cells are produced. Meanwhile, LH stimulates interstitial cells, also known as Leydig cells, located between the sperm-producing tubules, to produce testosterone. Testosterone is essential as it not only drives the development of male secondary sexual characteristics during puberty but also supports sexual drive and ensures that sperm can mature properly. Within the testes, a highly specialized structure called the seminiferous tubule serves as the site where sperm cells are generated from stem cells. As these cells progress, they develop tails and mature into functional sperm cells. Once formed, sperm cells aren't immediately equipped to fertilize an egg, they are transported to another structure called the epididymis. The epididymis is a coiled tube located at the back of each testicle, serving as both the site for sperm maturation and storage until ejaculation occurs. During ejaculation, sperm journey through the vas deferens and exit the body via the urethra. In the female reproductive system, these hormones play equally critical roles. FSH stimulates the growth and maturation of follicles within the ovaries, setting the stage for egg development. LH facilitates ovulation, the process by which a mature egg is released from its follicle. After ovulation, the remnants of the follicle transform, giving rise to the corpus luteum, a gland in the ovary responsible for producing progesterone. This hormone is vital as it thickens and sustains the endometrium, or uterine lining, preparing it for the potential attachment of an embryo. In essence, the corpus luteum is a key player in the early stages of pregnancy, as its hormones create a supportive environment for fetal development. If fertilization does not occur, the corpus luteum degenerates, triggering menstruation. If pregnancy occurs, the corpus luteum remains active, providing essential hormonal support. After ovulation, the egg makes its way into the oviduct, also known as the fallopian or uterine tube, where fertilization takes place. When a sperm successfully penetrates the egg, a zygote forms, representing the first single cell that contains the complete genetic blueprint for a new human being. This newly formed zygote quickly begins a process known as cleavage, characterized by rapid cell division that increases the number of cells without enlarging their overall size. As this embryo continues its journey towards the uterus, it must implant itself into the thickened endometrium to ensure its survival. Upon successful implantation, the embryo activates a critical mechanism and begins to secrete HCG, human chorionic gonadotropin. This hormone plays several vital roles. It sustains the corpus luteum, maintains progesterone levels, and prevents menstrual shedding, thereby securing the embryo's connection to the mother's body. Pregnancy tests detect HCG levels, which serve as a reliable indicator of pregnancy. As the pregnancy progresses, the hormone oxytocin serves an important role, triggering uterine contractions during labor and milk ejection during breastfeeding. Once implanted, the embryo embarks on a remarkable developmental journey, navigating through several key stages that transform a single cell into a complex body. The first of these stages is known as the morula, a solid mass of identical cells resulting from cleavage. During this early phase, fascinating occurrences can happen. Cleavage can lead to the formation of twins, which may be either identical, monozygotic, or genetically different, dizygotic. In later stages of cleavage, it's also possible for conjoined twins to form. Furthermore, if two moruli fuse, this can result in a rare occurrence known as chimerism, which has been observed in a limited number of cases in humans and is often the subject of biological research. As development progresses, cells exhibit remarkable flexibility, enabling them to contribute to more than one individual. Transitioning into the second stage, we witness the formation of the blastula. Approximately 5 to 6 days post-fertilization, the morula transforms into a hollow structure, which houses a fluid-filled cavity known as the blastocele. The blastopore, a critical opening in the blastula during gastrulation, ultimately forms the gastrula, which lays the foundation for the digestive system and serves as a precursor to both the mouth and anus. In humans, this blastula stage is called the blastocyst. The blastocyst is composed of two primary groups of cells: embryoblast, inner cell mass. This will develop into the entire fetus. Trophoblast. This layer forms crucial structures that support the embryo, including the chorion and later components of the placenta. The chorion serves as the outermost membrane that encases the embryo, providing initial protection as it attaches to the uterine wall and containing vital vessels for the exchange of essential nutrients and oxygen. It is important to note that successful implantation is a non-negotiable condition for pregnancy. If implantation fails, pregnancy cannot proceed. The placenta emerges as a remarkably complex organ that fulfills multiple functions. It supplies oxygen and nutrients to the developing fetus, removes waste products, acts as a barrier against some infections, and produces key hormones required for sustaining pregnancy. The fetus connects to the placenta via the umbilical cord, often called its lifeline, ensuring that the developing organism receives the nutrients and support it needs. As embryonic development continues, cells undergo a process of organization, leading to the formation of 3 germ layers. Ectoderm. This layer gives rise to the nervous system, skin, hair, and nails. Mesoderm. The mesoderm develops into muscles, bones, the heart, blood vessels, kidneys, and reproductive organs. And endoderm. This innermost layer becomes the lining of the digestive tract, the lungs, liver, and pancreas. This stage of development not only involves differentiation, in which cells specialize and acquire distinct characteristics, but also morphogenesis, the process by which organs begin to take shape and establish their functions. A phenomenon called neurulation occurs during this phase, during which a structure called the notochord, arising from the mesoderm, plays a foundational role in the development of the nervous system. As the notochord forms, it signals the overlying ectodermal cells to form the neural tube, which eventually differentiates into the brain and spinal cord. This intricate sequence of events marks a significant milestone in human development, laying the groundwork for the complex systems and organs that will emerge in the weeks and months to follow. As development continues, the growing fetus briefly shows features that reflect our ancient evolutionary past. Pharyngeal arches resemble gill structures before forming jaw and ear parts. A tail appears before shrinking into the coccyx. Eyes start on the sides of the head, then move forward, as in other mammals. These features disappear later, but they indicate that humans share a common ancestry with many other animals. Evolution leaves reminders, even before birth. As the embryo progresses through these stages, it continues to adapt and grow, laying the foundations for a fully developed human being. Each step in this extraordinary process is guided by a blend of genetic instructions and environmental influences, ensuring that the journey from a single cell to a fully formed individual is both precise and remarkable.